Maximize your data, copy number estimation from methylation arrays, next-gen sequencing, and SNP microarrays. Our presenter today is Dr. Andrea O'Hara, and I will hand it over to her so we can get started. Dr. O'Hara?
Thank you. So today I'm going to be talking to you all about how we can maximize our data. So what are the different sources of data that we can use for copy number estimation? How can we input them into Nexus copy number 8.0? And then what sort of results can we expect? So in Nexus copy number 8.0, the basic workflow is after we either open an existing project or create a new project, we're going to start with probe log ratio values. These are going to be copy number or copy intensity values based on the probes. From there, we are going to identify segments that have the same change. From those segments, identify regions of copy number alteration, and then within those regions, identify genes and functions that are going to be relevant for our analysis. The basic workflow is going to be the same regardless of which kind of copy number information we're using. However, there are a lot of different sources of data that we can use to estimate copy number. So what are some of the data sources that we can use for copy number estimation? Our primary methods are SNP Microarray, which is commercially available by Affymetrix, Agilent, and Illumina, as well as Array CGH, which includes both Agilent arrays and custom arrays. And these are what we think of as traditional sources for copy number analysis. We can also do copy number estimation using next-generation sequencing. This can be genome, exome, or targeted sequencing panel. And we can also use methylation arrays, which are available through both Agilent and Illumina as manufacturers. Again, with all of these, we can also use any sort of a custom array or capture as our source for the data. And we can use this for both constitutional or germline samples, as well as cancer samples. So my next questions are, though, with these different sources, how do these different methods work for copy number estimation? What levels of detail can we expect? And how do we load them into Nexus copy number? Our first example is SNP Microarray for copy number analysis. SNP arrays work by using short oligonucleotides to interrogate a single SNP. The signal strength from that probe is then used for copy number estimation. We can use this on constitutional samples or cancer samples. There's no requirement for a normal sample as it has a built-in reference control. So you can run just cancer samples, you can run constitutional samples with affected or with parentals or really any mix and match scenario that you want. The level of detail that we can see will depend on the specific array density. A more dense array will typically give us more information than a less dense array. However, most of the current arrays have a coverage such that small changes of less than 1 megabase can be easily detected in both exonic, intronic, and intergenic regions. That means we're able to detect regions that are going to be parts of exons or just small parts of promoter regions, intergenic regions, and intronic regions. And when we're using Nexus copy number 8.0, we're going to be able to directly load in either the .cell or the .os chip file for our Affymetrix data, Illumina final reports, Agilent feature extraction files, or a text file that includes probe information if we have a custom array. And so to do that, we would first launch Nexus 8.0 and either open an existing project or create a new project. And then we're going to pick our specific manufacturer here. So we'll pick Affymetrix, Illumina, or Agilent. We'll select our processing type. We're going to either select Mosaic for cancer or non-Mosaic for germline. Typically, when we're using the defaults, we want to slide everything over to stringent to begin with, as this typically yields the best results. And then we're going to check our perform systematic correction and select our specific array from the drop-down box. And then we'll load in our file for our results. And this is going to be the straightforward method for loading any SNP array. We can also use Array CGH for copy number. Array CGH works by hybridizing a known control DNA to an equal amount of sample DNA, and the comparison of the signal strength is what we use to determine copy number. We can run cancer samples or constitutional samples. Our cancer samples, for either sample set, we can use either a commercially purchased known DNA, or we can use a normal reference from within our own set. For cancer samples, sometimes we will use a matched normal for that individual person as a reference, but we can use commercial DNA here as well. And the level of detail here, again, is going to depend on the array density. Array CGH varies from being more and less dense. However, we're still able in most arrays to detect small changes of less than 5 megabases, and in the more dense arrays, less than 1 megabase. And again, the coverage is going to be spread out throughout the genome, so we'll be able to capture changes in both exonic, intronic, and intergenic regions. And when we load this into Nexus copy number, we're going to directly load either our feature extraction or a text file of our actual probes if we have a custom array. And so to do this again, we're going to either open an existing or create a new project. Then we're going to select our data type. This is typically Agilent feature extraction files if we're doing array CGH. We're going to select again if we're using cancer or non-cancer samples, slide it over to stringent, and then pick our specific array. And then we'll load in our files. So those were both of the more traditional methods that we use for copy number analysis, but we can also get copy number from some less traditional methods. The first is NGS, next generation sequencing, which can be genome, exome, or targeted sequencing panel. We can use our post-aligned BAM files and compare them between either a matched or a pooled reference to our samples in question. We can use this for both constitutional or cancer samples. With constitutional samples, we're going to want some sort of in-batch normal. This can be a set of samples that are known normals that we're sequencing within that batch of samples, or we can use the entire pool of the constitutional samples to serve as a reference that we can then compare each one individually to. For cancer samples, we can use an in-batch matched or unmatched normal. This means that we can have tumor samples that have the same individual's blood or adjacent tissue sample as a matched reference, or we can have it as an unmatched if we have other sources of DNA. Again, we would prefer this to be, though, an in-batch sample. The results in terms of our level of detail are going to really depend on several different factors, including our genomic coverage, our sequence depth, and the overall sample quality. We would expect that whole genome sequencing, we're going to really get the entire genome for coverage. So we'll have both introns, exons, intergenic regions, the whole range. Exome is really going to be targeted just to the exons, so we are not going to have any capture or any changes that are in intergenic or intronic regions. And targeted panel will typically offer us very rich level of detail, but only for specific genes. This may cover both introns and exons within those gene regions, but it is going to be targeted to a specific set of typically 50 to upwards of 500 genes. Our sequence depth is also going to impact how well we are able to have our results, and that's going to be reflected also in our sample quality. Typically, the deeper that we go, the better our results are going to be, as we're going to have more capture and more to work with. And when we load this into Nexus copy number, we have two different methods that we can use. If we have constitutional samples or unmatched cancer samples, we're going to pre-process these files and then load it into Nexus. If we have matched tumor normal pairs, we're able to load the BAM files directly into Nexus. And so to do that, if we have the genome, exome, or targeted panel, and we're looking at either a constitutional sample set or an unmatched tumor normal sample set, we're first going to launch the BAM Reference Builder. This is an app that's located within your Nexus 8.0 installation folder. We're going to be running this before we launch the software. And when we run this, we're going to pick out our organism and build, our region file, which is going to specify our capture. This is really important if we're using a targeted panel, as it's going to limit our results to the capture regions so we don't have any off-target hybridization noise contributing to our results. If we're using exome or genome, you can add the region file, but it's not necessarily required, and the default will switch to being about 100 base pair window. From here, we're also going to select our BAM files. Again, this is going to be our already aligned files of our FASTA and FASTQ, so we have aligned read depths. We prefer 10 to 20 samples to create a reference. You can use less if you have it. After a certain point, you can use more as well, but after a certain point, we do find that it just takes longer to run, but we're not necessarily getting additional information. So 10 to 20 is the optimal range. If you only have one sample to use for your reference, you're not going to want to use the BAM reference builder because this is, again, creating a pool. So if you have one sample, you're going to want to use the next method, which is the match paired. After you've created your pool of 10 to 20 samples, you are then going to either, again, open an existing project or create a new project, and then load your samples as the data type BAM pooled reference. You'll specify your reference pool that you created from that app, and then load in your individual BAM files here. For a constitutional data set, we may choose to make a pool of all of our samples, and then we can load each sample individually here. If we have a tumor normal set, we would make a pool of our normals, and we can either load in just the tumors or the tumors and the normals if we want to see how our normals also compare. The choice is yours in terms of your options for that. Now we also may have genome or exome data or even targeted panel where we have a matched tumor normal sample. That means that we have the same individual with two samples, one that's going to be in their tumor and one that's going to be either a tissue or blood normal. In this case, we want to load those BAM files directly into Nexus using the BAM NGCGH matched. We will, again, open our project and then load in our tumor samples as an experimental and then the exact matching normal over here. If we have only one normal reference, we can do the same idea here where we're going to match each sample individually to that one normal. And this is ideal if we're going to be doing an exact pair analysis. Finally, we can also use methylation to do copy number analysis. So methylation arrays work using probe intensity values as compared between either a matched or pooled reference samples and the sample in question. This is sort of a combination of using both the array and the pooled reference from the sequencing. We can use both cancer or constitutional samples. For both of these, we do require some normal samples also run on the array. Tissue-specific is preferred, but not required. The level of detail in the methylation microarrays is not as uniform or as dense throughout the genome as the other arrays are. They typically have less probes than the SNP and array CGH microarrays, and also these are going to be located primarily at promoters and CPG islands, so the coverage is not going to be quite as uniform throughout the genome. However, we can still reliably call changes of greater than 5 megabases, and even more so for larger changes, so things that are going to be parts of partial chromosomes, chromosome arms, or full chromosome changes. And to use this in Nexus copy number, we're first going to pre-process our raw image files using typically a program in R, and then we'll load either our probe or segmented results. In my testing of various algorithms, I found that Kanume worked best for the methylation results. So Kanume is a package that is available on the Bioconductor website. I have the full URL up here at the top. This is an algorithm that can be used to determine copy number variation from the Illumina 450K methylation array. This vignette right here gives a very detailed step-by-step instructions of how to load in your data and get out a text file that you can then use for copy number analysis. This particular tool uses a raw.idat file, which is basically the image file. This is the rawest information that we can get from our methylation arrays. It's going to take in that information. We are going to specify a set of 10 to 20 samples for the reference. In the examples that I'm going to show you today, I used anywhere from 12 to 24 samples for my references. You're going to specify reference samples and then input your other samples that you want to estimate copy number from. The output is a text file, and for this particular algorithm, it's going to be segments. I'll get into that in just a moment. Once you've exported your results from this package or another, you can upload your results directly from the output into Nexus Copy Number using your custom data type. For this one, that custom data type is MMRC, and I'm just going to be selecting my text files and loading them in. So the basic workflow is going to be as follows. For our SNP array or array CGH, we are going to be loading in our raw data at the level of a probe log ratio value. So all of the probes are going to be loaded in individually, and we'll have log ratios for each of these values. It's the rawest data that we can get to upload into Nexus Copy Number. NGS is also going to be loading in as log ratios, and it's something similar to the probe level, but slightly different because it's going to be based on our capture. So these probes are not going to be as small or as fine as the SNP arrays or array CGH. They're typically going to be on the order of anywhere from maybe 100 to 500 bases, depending upon our capture. So slightly larger regions, but still a very fine level of detail. The methylation data, on the other hand, may be in the probes or as segments, and this is going to depend on which package you choose to analyze your data with. Again, I tried several different packages, and I found the results best with the Kanume algorithm, which gives us segments as our output. Just to illustrate what this is going to look like, I have an example of the same sample with two different data sources here, one on the left that's showing us probes and one on the right that's showing us segments. So if we look over here on the left, we're zoomed into an area near the centromere. We can see here each of these individual little dots is a single probe. So we have probes scattered all throughout the region. We also have these thick black bars. These are the segments that are being drawn from the probes. And then depending upon where they're falling in this spectrum, we can see if there's a copy number change. So right here, this segment is underneath this thin red line, indicating that we have single copy loss in this region. On the right, we have the same sample. Now, this is only showing us the segmented data that was loaded in. We do not see the individual probes loaded here. We just see these thick black bars for the segments, but we can clearly see that this segment has fallen below the red single copy loss threshold, so we have loss here. And so while these fine details look different when we zoom in to this particular view, if we look at the output of our results, we see the same overall result, that we have loss of this region. So even though the details are varying in terms of how fine of a detail we're seeing, we're going to really still traditionally get the same output for our results. So I'd like to take you through two different case studies to show you how these data are actually working in sort of a real-world environment, what we can all see, and hopefully show you that you can use any of these data sources reliably in Nexus Copy Number for your analysis. Because methylation is sort of a newer source for copy number data, this first case study is a proof of principle study. We have a series of normal and trisomic placentas from chorionic villus samples, or CVS samples, as well as maternal whole blood. All the samples were subjected to FISH analysis as well as DNA methylation profiling with the Illumina 450K human methylation bead chip. As I mentioned, the Illumina methylation results were processed by Kanume as instructed in step-by-step tutorial on their website, and the CVS samples were compared to whole blood results. I actually did two separate comparisons because there were both normal placentas as well as this maternal whole blood. The normal placenta is going to be a tissue-matched reference, while the whole blood is not tissue-matched. I found that when I used either set or both sets together, so either 12 normal, 12 maternal whole blood, or all 24 normals, I had the same output result for our trisomic placentas. So I'm showing you the results today of the comparison with just the maternal whole blood. So let me just show you what this looks like. I'm going to launch Nexus Copy Number. So I already created this project so we can see the data already loaded in. So we'll go ahead and open the project. I'm going to take you through the steps of how we load in the data and then what this data looks like when we view it. So to get started with the methylation data, again, I pre-processed it in R package using Kaname. R is a software program that's freely available. If you just Google R software program, you can find it and download it. And then the Kaname software package is also freely available on Bioconductor. You can download that, install it into R, and then process your IDAT files right through it. It's a very simple and straightforward process. Once it's been processed, that primary output, which is a .seg file, can be loaded into Nexus copy number. So we're going to go to load data, and we'll select our data type of MMRC. If you're unsure of what your data type is, you can always contact us and we can help you find the correct data type to load it as. We'll select the files, and then here I'm going to just load these .seg output files directly into Nexus copy number. Okay, I've already loaded these files in and processed them so we can view them. So I'm going to go ahead and click View so we can see the results. I'm going to add a factor here of our CVS FISH results. Again, these were already processed by FISH, so we already knew what the result would be. And so this is a way for us to confirm that we are seeing what we expect. So on the top here are all of the samples that had a normal FISH result, meaning that there were no gains or losses of large chromosomal regions. This test was looking for trisomy of 13, 18, and 21. You can see that we have a few very small changes. These could be germline changes or it could be artifacts, but we're really unable to tell based on this particular information as we only have FISH analysis. However, we can see that 13, 18, and 21 are completely clear. If we scroll down, we can see here that on chromosome, we have a series of samples that have a gain of chromosome 21, so trisomy 21, as indicated by this blue bar on each of the samples. And again, if I go over here, I can see that our CVS FISH result for these was trisomy 21. If we scroll down further, each of these individual samples is showing gain of chromosome 18, as indicated by the blue, or trisomy 18. And then our last set here, we have several samples that have trisomy 13 right here. If we want to view these as aggregates, I can go over here. Let's just zoom in a little bit to see more clearly. We can see that all of our normal samples are relatively normal, with perhaps a few small changes that are most likely normal germline variation. Our trisomy 13, all of them show gain of chromosome 13. Trisomy 18, all of them show gain of chromosome 18. And trisomy 21, all of them are showing gain of chromosome 21. And again, we do see a very small number of other small calls which may or may not be there, but based on our results, we can't validate at this point. So this data set showed for me that we can reliably call at least large-scale changes in samples using these files. My next question, though, was to ask, what is the level of detail, and how is that going to compare to some of our other methods of data sources? And so to check that, I decided to go back over and look at a second series of samples. So case study number 2 is using breast cancer, or BRCA, tumor and normal samples. These samples were subjected to SNP microarray, DNA methylation array, as well as whole exome sequencing. For the SNP array, we have matched tumor and normal samples as processed by an Affymetrix SNP 6.0 array. For the whole exome sequencing, we have a read depth of approximately 25x coverage for both a tumor and normal matched samples. And then finally, we have a series of tumor samples as compared to unmatched normals, approximately 16 samples that were run through an aluminum methylation array and then processed by Kaname. And so I'd like to take you through these results in Nexus copy number. So to start off, I'd like to show you what our traditional microarray looks like. As I mentioned, microarray is what we typically and traditionally use for copy number analysis. And so this is what many of us are both familiar with and what we typically set as the standard for copy number assessment. We're just going to be looking at a small cohort of 3 samples, but we're going to be looking at these in depth across all of our different data platforms. So To load this data in, we're going to go over to our Load Data menu and pick our specific manufacturer. Here, this was the Affymetrix cell data. So again, I would be picking Affymetrix cell. These were cancer samples. Slide it over to stringent, and then I would pick the SNP 6.0 array and load my arrays in. Now, if I take us back over to our results, these are already processed. And we can see here our copy number changes. So we have red for loss, blue for gain. The top is showing us our aggregate or summary profile, and then we have individual profiles for each of our 3 samples. I'm going to go ahead and add a factor that's showing our data source, so we're able to keep track as we're going along. All of these yellow ones are going to be indicating that we used a SNP array. You can see that in addition to having our copy number profiles here, we also have B allele frequency information. That's because the SNP probes do also offer us allelic information, so we're able to see if we have any areas that have allelic imbalance or loss of heterozygosity, changes in the allele frequency. You can see that we have both large changes covering chromosome arms, whole chromosomes, large portions of chromosomes, as well as some smaller changes that might be on the order of several kb or megabases. And they really vary widely in terms of their size. Okay, we can also now add in our methylation data to see how it compares to this standard array data. So if we go back over to our data set, I'm going to select our methylation arrays here. And so in order to load the methylation data, again, I click on Data, I went down to MMRC, and then I selected my direct output from Kaname, which is these .seg files. And if I load these in, okay, you can see now the arrays are shown in blue, and then the methylation is shown in red, or sorry, in yellow. Again, we have red for loss and blue for gain. You'll see that when we look at the first sample, we're able to clearly detect both these small changes here, which is approximately 4 megabases, as well as these larger changes that are covering chromosome arms. All the way throughout, our methylation results are only giving us copy number results. We will not have B allele frequency, and we're not able to detect some of these smaller changes. Now again, this could be due to differences in coverage between the 2 platforms because our methylation array is really focusing on those promoter and CpG islands. So if it's an area that's not covered by a promoter or CpG island, we won't be able to detect it. In our second sample, we're again able to detect all of these large changes. But again, there are some small changes that we're not seeing. In our third sample, we can see we can clearly detect these gains, but it looks as though it's missing some of these losses. And my next question when looking at this is to see how does the whole exome sequencing compare across each of these 3 samples, especially sample 3 in particular. So I'm going to go ahead and select our whole exome sequencing results. Again, these are matched paired samples, so to load these in, we are going to use the BAM NGCGH method, which allows us to do a pair-matched analysis of a tumor versus a normal control. If I did not have any normals that were related or I wanted to create a pool, I could do that as well. But if I have matched samples, direct one-to-one is typically going to be our best result if I have good quality results. And again, these were all sequenced at a fairly good depth of 25x, so we did have a nice coverage for the genome. I'm just going to zoom in. Okay, so we have the array in pink, methylation in teal, and whole exome sequencing in yellow. You'll see that for this particular method of using NGCGH, we are going to see just the copy number. If we use a pooled reference builder, we can also incorporate B allele frequency data as well. So right now we're just getting our copy number. You can see that it's reliably calling both the small and large areas. There are a few small changes that are different between these sets, but overall they are extremely similar for sample 1. Sample 2 likewise is capturing these big changes quite well. We can see that for sample 2 it does seem to be capturing some other areas of loss, and this could be due to differences in coverage or it could be due to reduced sample quality. For the third sample, you can see that again we missed these losses for our methylation arrays. And we can see that they're kind of peeking out in some of the whole exome sequencing, but the coverage, again, is not what we would hope for. So one thing that we can do is we can take a closer look at our raw input data to see if that gives us any more information as to what's going on here. So if I click on our sample name over here at the left, I can do a single sample drill down. And if I go to the whole genome, I can actually see here our results. So again, we're looking at the segment values. These thick black bars are our individual segments. 0 is our baseline. This blue line right here is single copy gain, and this red line is single copy loss. So you can see that we have areas that are above the single copy gain threshold and are being called as gain. We also have some areas that are below the baseline, but are not below the single copy loss threshold. So there are areas that look to be lost, but because they haven't cleared this threshold, we're not calling them as such. If I click on the whole exome sequencing to see what's going on in that sample, you can see that unlike with the methylation data here, we have probe level data, not just segments. And so we can see clearly these areas that are gained above the blue bar right here and right here. We also see areas that are definitely dropping down. I'm going to zoom in so we can see a little more clearly. So we have areas that are dropping down below the baseline here, but they're not all the way down past the threshold. So it appears that in this particular sample, the DNA that was used for the methylation array and the whole exome sequencing, the percentage of tumor sample was not quite as rich, so we're unable to see all of the losses. This is more of a mosaic sample. It has a higher level of contaminating normal. But what we can do is we can very easily adjust this threshold here and here, adjust these thresholds to help capture that loss. So what we can do, I'm going to do it for the methylation sample. We'll go ahead and go to our settings menu, and I'm going to select the MMRC data type, and I'm going to adjust the loss threshold. Right now it's at 0.3. Our losses were at roughly a little bit less, or around 0.2. So I'm going to go to negative 0.15 so I can accurately capture those changes. I'm going to click Done. You can see my settings have been saved. Now, right now, nothing is going to change because all of these samples have already been processed. But if I highlight the sample and reset it, you can see it's now unprocessed. And when I click View, it's going to reprocess it with those new settings. And if I zoom in here, you can see now my third sample, this is BRCA3, I'm now able to capture all of these losses that we were seeing in the array also in the methylation array. And I could do the same thing likewise with our whole exome sequencing result to capture those losses as well. So hopefully I've shown you that you can actually use any of these three data sources reliably as a way to do copy number estimation. Now, depending upon the data that you have, you can use, you can mix and match these for your different samples. If you have all three sources, you can do copy number estimation from all three. The one thing you'll need to keep in mind when you load in your data is that they each have to have a unique name. Here I've just renamed them based on their format, but they do need to have a unique identifier. If you have only one for some data source, only one source of data for some samples and another for the other, that's fine. You can mix and match. Just keep in mind that you do have differences in coverage, so you may not always detect very small, fine changes with each of these different methods. Now, in addition to extracting copy number information from these sources, we can also integrate our alternative data sources into our analysis using their traditional output. So if we go to our main data set tab and go to the data loading guide, we do also have the option of loading in sequence variant data from NGS right into our individual samples. Again, this sample name, you'll want it to match your name over here. If you have replicates, you can load in multiple copies and just adjust the name. But this is a way for you to load in the sequence variants, so your all of your single mutations, small indels, changes in stop, any sort of splice site information that you want to include. You can load that in for your VCF through a VCF or MAF format. Additionally, if you have expression results from either expression microarray or RNA-seq, you can load in your individual sample expression results here. Likewise, if you have a group-wise expression, you can upload your differential expression results here. And as we were just talking about methylation, you can also upload your methylation results here for differential methylation regulation. So you can really use your data in both its primary focus and as an alternate source for copy number, right all inside of Nexus copy number. And so with that, I'd just like to draw your attention to some additional resources that we have. If you're looking for more information, if you'd like to see some additional webinars to show some of the downstream functions that you can do with your data once you have it, some educational videos that are going to further describe and detail some of the features I might have glossed over in the introduction, or additional resources, you can go to our website at biodiscovery.com/resources. And of course, if you have any questions about this content or anything else, you can also feel free to email us directly at support@biodiscovery.com.
We will now move on to the audience questions.
So the first question is, when is Nexus only using log2 ratio data, and when is it using B allele frequency data as well? So the answer to that is it depends on which data source you're using. So for the SNP Microarray, you will have both log ratio information for copy number as well as B allele frequency for LOH and allelic imbalance. With array CGH, you will only have copy number information, so only the log R values. With sequencing, it will depend on which algorithm you use. So the BAM pooled reference does include B allele frequency calculation, while the matched paired NGCGH does not. So that one is, whether or not you have that information will depend on which algorithm you select. For methylation data, that is only going to include the log ratio value and not the B allele frequency. So depending upon what source you have and which way you process it will tell you which information you have. Okay, the next question is there any way to change the baseline in the samples? All of the samples that I showed you today for both the constitutional and tumor samples were using their default median recentering. They did not need to be recentered. However, with tumor samples, we sometimes find we do need to recenter them. With any of these methods, if you find that your most common area, which is likely your baseline, is shifted, you do have the option within Nexus copy number to adjust the baseline from our whole genome view. This is true for all of the different inputs that once you've put it in, you can make that adjustment as needed. Okay, the next question is, are all the methylation data you showed today Illumina 450K array data? And the answer is that yes, today I did use that because I was just sticking with one algorithm. Depending upon which array you use and which manufacturer and which array, you may choose to use other algorithms. There are a lot of different algorithms that are good for different platforms. I found that for this particular data set that I had, Kanume was the best algorithm, which was using these 450K Illumina arrays. All right, the next question is, sorry, I'm just reading ahead. It says, how is constitutional mosaicism dealt with? So the constitutional mosaicism as well as cancer mosaicism are always an issue. Typically what we're going to do is start with our default cutoffs for the threshold values, but it's always good to take a look at the single sample view to see if you're capturing all your changes. We saw with that one cancer sample that the methylation and whole exome sequencing was not fully, the thresholds were not fully capturing the changes in those two data sources. Likewise, the same can happen with our constitutional samples as well. The general rule of thumb is that we're usually reliably able to detect changes that are occurring in at least 30% of cells. If it gets to be lower than that, it can be quite difficult to determine whether or not any changes are happening. But the best way to see if you're capturing those changes is to actually give it a quick look and see if you're seeing any segments that are falling in between our baseline and the threshold cutoff. Okay, what is the ideal coverage of whole exome and targeted sequencing data for copy number analysis? That's a really great question. So in general, a deeper coverage is going to be, a deeper sequencing is going to be better. So in terms of the percentage of genome, whole exome is going to typically offer us more detail than targeted sequencing, and the deeper the sequencing goes, the more reliable our results are going to be. For constitutional samples, you don't necessarily have to go as deep as you do for cancer samples to catch reliably all of the calls. And this again has more to do with the mosaicism of the sample. But typically something that's on the order of 20 to 50x is going to be great for doing copy number analysis. There are ways to use low-pass reads, but that is less reliable right now, so we typically recommend using deeper pass information for your NGS results. This is a question that's come up for a few people. I noticed that you adjusted your thresholds on just one sample. Should you apply the same criteria for all samples, and how do you decide what that criteria is? We were using a very small data set of only 3 samples, so it's a little bit trickier in a very small set such as this, but I typically find that I like to start with a one-size-fits-all approach and then adjust as needed. What you'll find in both constitutional and cancer sets is that the same settings are most likely not going to be appropriate for every single sample. So what I like to do when reviewing the samples is I usually can start with one setting that's going to fit most samples and then use one alternate setting that should capture the changes in the remaining samples. This is typically a lower level mosaic setting, so capturing those changes in the samples that have a slightly lower affected cell population. I do think it's always important if you're using alternate settings, though, that you do make a note of those samples and then refer to that, when you're writing up your methods or presenting it, so that everybody's aware that you have used alternate settings. But it's perfectly acceptable to use more than one setting as needed to best reflect the population of cells that are affected in your samples. Okay, and let's see, I'm going to take just a couple more questions here. Okay, so sorry, I've gone over some of these. So for the methylation data, I do recommend using Coname if you're using the 450K methylation array. I found that worked best for the different methods. The question was, which algorithm do you recommend? Again, if you're using other platforms or other arrays, there are other algorithms that will function better, but I can't quite give you the full chart layout of all of the different options because there are quite a few. These are all based on array and not based on bimethyl sulfate sequencing. I'm not actually as knowledgeable as in terms of copy number estimation from that method. Okay, again, another question is, with Coname, you only get the segment data but not probes. What program would you want to use if you want to get the probe-based data? CHAMP is another program that's very similar to Coname but will offer you probe-based information. But I did find that it wasn't quite as reliable as Coname in my analysis. Okay. And with that, I think we'll take it over to our ending questions. If you have a question that I didn't answer, just please know that we'll get back to you after the presentation. And Shalini, if you would like to take it away.
Thank you very much for a very nice presentation.